student of PharmD 5th year, and I am Vatsalya, student of PharmD 5th year. Welcome you all to the International Conference from Workshop, Alternatives to Animal Experiments, Exploring Imminent Possibilities in Preclinical Drug Discovery using Computational Tools, that is AAE 2023, organized by Amity Institute of Pharmacy, Amity University, Uttar Pradesh, Lucknow campus. Science advances not by tradition but by transformation. Embracing computational alternatives in drug discovery is our beacon towards a more human and innovative future. The need for alternatives to animal experiments in preclinical drug discovery is driven by ethical concern, cost and time saving, improved human relevance, reduced variability, environmental impact reduction, and increased regulatory acceptance. By embracing computational tools, we can create a more ethical, efficient, and sustainable approach to drug development. AAE 2023 is an immersive and thought-provoking conference and workshop aimed at students, PhD scholars, educators, researchers, and professionals in the fields of pharmacy, life sciences, biomedical research, and related disciplines. This program is designed to explore and promote ethical cutting-edge alternatives to traditional animal experiments, fostering a deeper understanding of the latest advancements in non-human research methodologies. It further aims at providing hands-on training to the attendees on handling different freely available computational tools useful in drug discovery and development, including orthodox, Swiss ADME, etc. To grace this inaugural ceremony of this international conference cum workshop, AAE 2023, we have with us Dr. S.K. Ratsar, Chief Scientist, Division of Toxicology and Experimental Medicine, CSIR, CDRI, as the speaker of Technical Session 1. We are also privileged to have with us Dr. S. Ankomani Sir, Principal Scientist, React Division, Ecotoxicology Laboratory, CSIR, IITR, Lucknow, as the speaker of Technical Session 2. We have our Honorable Wing Commander, Dr. Anil Kumar Sir, Deputy Pro VC and Director, SN, AUUP, Lucknow campus, and Dr. Kamar Rahman Adam, Distinguished Professor, Dean Research, SNT, AUUP, as the chairperson of AAE 2023. We are also privileged to have with us Professor Dr. Rajesh Tiwari Sir, Dean Academics, Director Outcome, FPOC Swayam NPTL, AUUP Lucknow campus with us. Last but not the least, we have among us our beloved Professor Dr. Anuradha Mishra Madam, Director, Amity Institute of Pharmacy, AUUP, Lucknow campus, as the convener of AAE 2023. To make this day blessed, I would like to invite our esteemed dignitaries and the organizing secretaries of AAE 2023, Dr. Shraddha Bist, ma'am, Associate Professor, Department of Pharmacology, Dr. Lucy Mohapatra, ma'am, Associate Professor, Department of Pharmacology, Dr. Rahul K. Maurya, sir, Associate Professor, Department of Pharmaceutical Chemistry, and Dr. Mohammad Yasir, sir, Assistant Professor, Grade 3, Department of Pharmacology, AIT, AUUP, Lucknow campus. So please join us on the stage and inaugurate this event by lighting the sacred lamp.
दूसरा लैपटॉप है सडनली वही सिंक करवाया कल चेक बहुत अच्छा व्यू चला था देखिए थे अभी एक घंटा वो सोच रहा था 6 बजे करना चाहिए
जाऊ दे बाहेरची लक्ष्मी घरात येऊ दे घरच्या सर्वांना उदंड आयुष्य सुख समाधान शांती लाभू दे
निकालना नहीं तुम्हारा पोर्ट भी जाएगा और हमारा ये नहीं सर वो बिना बताए मत दोनों
Thank you, one and all. May Lord Ganesh bless us and empower us to conduct this noble event flawlessly. I would now request Professor Dr. Anuradha Mishra Madam, Director, Amity Institute of Pharmacy, Amity University, Uttar Pradesh, Lucknow campus, the convener of AAE 2023, to present the welcome address.
Warm greetings, and it's a very blessed day. Very good morning to one and all. Honorable Deputy Pro Vice Chancellor Sir, Wing Commander Anil Kumar Sir, our esteemed Professor, Dean Research, Science and Technology, Dr. Kamar Rahman Ma'am, and Dean Academic, Professor Dr. Rajesh Kumar Tiwari Sir, and Dean and Director AIB, Professor J.K. Sir, and Shalini Ma'am, Director. I just give you warm welcome. Other than that, I can extend my warm welcome to our reverse speakers of today's technical session, Dr. S.K. Rath, Chief Scientist, Division of Toxicology and Experimental Medicine, CSIR CDRI, Lucknow, and Dr. S. Anupamani, Principal Scientist, React Division, Ecotoxicology Laboratory, CSIR, IITR, Lucknow. All esteemed dignitaries, faculty members, my dear students, and those who have joined us on Zoom platform. I extend a warm welcome to everyone to these premeditated animal experiments which we are planning for today's conference, international conference come workshop on alternative to animal experiments. Exploring imminent possibilities in preclinical drug discovery using tools. Organized by Amity Institute of Pharmacy, Amity University, Uttar Pradesh, Lucknow campus from 1st to 3rd November 2023. Being the convener of this wonderful 3 days international conference in hybrid mode, I would like to compare the primary objective of this conference come workshop is to ensure a platform for researchers, students, academicians, faculties to engage in meaningful discussions about alternatives to animal experiments. The vision of this event to support research by following 3 R's principle. This 3 R's principle of animal use says reduce, refine, and replacement. In our today's speakers will better deal with this subject, how we can reduce, refine, and replace the animal experimentations by using alternative tools, either in the simulation mode or the computational tools which is available with us. Several researchers suggest that non-animal testing methods such as using cell culture techniques in human and cell tissue-based testings, human volunteers testings, simulation techniques using computational tools. These are some alternative methods which you can prefer rather than the animal experiments. Advantages of these alternative tools are reproducibility, accuracy, cost effective, and quicker then and easier also if we are comparing with that of your traditional or conventional animal experiments. Many alternative to animal testing methods aim to overcome translational barriers towards developing urgently needed requirements like untreated ailments, like some conditions, some disorders which are not treated or still there will not be any available treatment regimen or therapeutic regimen is to these particular diseases. Without sacrificing quality and safety, alternatives to animal testings would provide significant outcomes in this field. Further, this great event aims at enabling PhD scholars and students of master in pharmacy, masters in biomedical domain from diverse disciplines to explore the profound connections between the inner facets of alternatives to animal uses and the different domains of science and technology. I am excited to announce that the MIT Institute of Pharmacy at MIT University, Uttar Pradesh, Lucknow campus is hosting this multidisciplinary international conference come workshop, AAE 2023. And the theme of this event resonates strongly with the vision of values set forth by our Honorable Founder President Sir Dr. Ashok K. Chauhan. Aimed at making MIT University a global educational hub with holistic approach. These 3 days international conference come workshop will also provide an insight on handling free softwares related to molecular docking, Autodoc, Swiss, and ADME. These are the softwares on which our faculty members will train our students. I'm glad to mention that an oral presentations competitions will be organized for researchers to exchange their scientific viewpoint, and the base presentations will be awarded with cash money. I am privileged to announce that we have received total 539 registrations from various states of India, such as UP, West Bengal, Gujarat, Odisha, Jharkhand, MP, Uttarakhand, Tamil Nadu, Telangana, Maharashtra, and Haryana, and other countries including Saudi Arabia and Nigeria. Almost 350 delegates will join offline on 3rd of November to attend this workshop. I am glad to share that we have received almost 141 abstracts for oral presentations, which has to be held during the post-launch sessions tomorrow. I extend my heartfelt appreciation and congratulations to all my organizing team members, student volunteers, faculty members for organizing this international conference come workshop on appealing theme, and my best wishes to all my participants, those who have joined online as well as offline. Thank you, one and all. Jai Hind.
Thank you, ma'am. May I now have the privilege to invite onto the stage Dr. Kamar Rahman Madam, Distinguished Professor, Dean Research S&T AUUP, to address the gathering online and offline as the chairperson of AAE 2023.
Good morning to everyone, and Dr. Tiwari head up this Lucknow campus, all the dignitaries from outside and from our institute, the staff members, and all the students. I'm very happy about this conference because it's a very important subject, and this subject, there was a time It was very close to my heart. And my 3 students at Rostock University, Germany, are working on this subject only. Dr. Shailen Gupta, Dr. Suji Gupta, and KP. KP has done his PhD from here. And at the moment, why I say it's important? Because number one, ethically using animals is not very good. Number two, they are costly, they are time taking, and we have to spend so much money also, as I said. Third thing is that we want quick results. You know, during the time of COVID, we developed drugs, but we wanted to save the safety. And safety is very important. I remember when I was working on the toxicity of nanomaterials, I used to do cell culture, human cells, and other things. That time I was not aware about much, although my students were, and that time I read also, now I forgot those other things, about the computer methods using the safety of the drugs. At the moment, FDA, Food and Drug Administration from USA, is giving lots of importance on the use of artificial intelligence. And they say that it helps a lot. It's quicker, safe, and it gives good results. And during the time of COVID, they gave lots of favor or lots of help to develop the artificial intelligence method for the safety of the drug, for quick development, all those things. And it's not very costly also. So at the moment in United States and India too, they are working a lot. I will be very happy the day when you people develop some method and do it quickly. It's a very important subject because drugs comes. We have to see that how they behave in human body. And when you know how it is behaving in human body, how it goes and what happens, it's very, very important. Because some of the drugs which came during the COVID time are not very good for human body. Although they were tested, but I think they were not tested properly. And there is also one thing that animal testing is important. But if you can do other testing, I think animal testing is not important. And I wish all the best because the experts are there, let them talk about this subject. And I always say one share when I give a talk. And the share is, Jala sakro to jalao arzu ke chara. शहर की राह न देखो शहर मिले या न मिले। जब ये हम लोग बत्ती जला रहे थे तो मुझे ये शेर याद आया। मुझे लगा कि ये शेर बत्तियां जलाई हैं हमने, ये अच्छा सिम्टम है। थोड़ा पोल्यूशन जरूर होता होगा लेकिन ज्यादा नहीं, थोड़ा सा धुआं निकलना। लेकिन ये क्या सिग्निफाई करता है? ये सिग्निफाई करता है कि आप शहर की राह पर देखिए, अपना काम कीजिए। And if you can come up with something, because आप ही लोग कर सकते हैं, the young generation should do it, then please do it and see. You have the facilities at the moment. Lucknow is a hub of science and technology. You can develop your collaborations with IITR, with CDRI, with CMAP, with Botanical Research Institute. Please do that. And we are also going to develop another MOU with medical college also, because for the first time in the history of King George's Medical College, Dr. Sonia Nityanand, a very able person, has become the Vice Chancellor of that university. So I wish you all good luck and I congratulate the organizers for organizing this very nice conference. I will not call it international, but it is partially you can say international, but it is a good conference. Thank you very much. I always avoid to come on the stage because when I come on the stage, you people can guess my age. Otherwise, it's fine.
Thank you, ma'am, for gracing the podium. I would now request Deputy Pro VC and Director Asset, Amity University, Uttar Pradesh, Lucknow campus, the esteemed chairperson AAE 2023, to join us on the stage to present the inaugural address and enlighten all of us with his enthusiastic words.
Our esteemed guest this morning, Dr. Rath, Dr. Ambumani, my esteemed colleagues and senior Dr. Kamal Rahmanji, Dr. Tiwari ji, Dr. J.K. Srivastava, and Dr. Anuradha Mishra, Dr. Shalini, and all my friends and colleagues. A very warm good morning and my felicitations on this international conference cum workshop on alternatives to animal experimenting and exploring imminent possibilities in preclinical drug discovery using computational tools. An extraordinary subject and very apt subject. But at the outset, I would like to extend a very warm welcome to Dr. Rath and Dr. Ambumani for having spared their valuable time and for being with us. So this warm welcome is not on the behalf of just Amity University Lucknow campus, but on behalf of the entire Amity universe, headed by our Honorable Founder President, Dr. Ashok K. Chauhan, whose vision is to make India a knowledge superpower. This warm welcome is also on behalf of Dr. Atul Chauhanji, who's the Chancellor of the University, Dr. Aseem Chauhanji, who's the Chairman and Chancellor of Amity University, Haryana and Rajasthan. This warm welcome is also with the blessings of Honorable Vice Chancellor, Dr. Balvinder Shukla, an academician par excellence. And I would also say this is such an auspicious occasion and this is such a good subject to talk about on the day of Karwa Chauth. So a very happy Karwa Chauth to all the ladies who are celebrating this great festival. This festival is about compassion. It is about human values. It's about goodness of relations. And that's what all avoiding animal experimentation is all about. I don't know how many of you have seen a movie called Project X. Has anyone seen this movie? Please raise your hand. Okay, in case you haven't seen, you should Google and see this movie called Project X. It's about mammalian experiment on experiments being conducted by the US government on effect of nuclear radiation on cognitive function of mammalian brain. So what do? The experiment is carried out on monkeys. And this monkey is subjected to fly a simulator. The highest level of cognition and motor skills are required in the flying of an aircraft. So this mammal is trained to fly objectively an aircraft. They give him inputs and give him the chocolates so that he flies the aircraft straight. Then they put nuclear radiations on top of him. And they find out how much time does it take for the cognitive function to decline. Now what happens is, as soon as the mammal passes the first grade of test, they put a yellow collar. When it passes the second grade of test, they put a blue collar. And third time, when he becomes a super cool pilot, they put a red collar. And red collar is also an indication that the animal is ultimately going to be sacrificed. In the sense, the radiations are going to be so strong that the mammal flying the simulator would ultimately die. And they would see how much radiations would actually affect in a nuclear war to a human pilot. You know, there's a mammal, there's a monkey who's very intelligent and he has undergone, sign language. He gets into the habitat where all animals are caged. He teaches sign language to all of them and tells them about the experiment. And one fine evening, they all escape when their head is about to get a red collar. And what this mammal does is, he takes all of them on the plane and flies them away. It's a beautiful movie, but the message is loud and clear that we must have compassion for animals. This topic is so close to my area of work, my thought process, that I was mentioning to a veterinary expert I was talking. He said, we're developing such a computational model, we're working towards it. And he said, sir, I'm very happy because in Lucknow, there was business of, he was saying, hundreds of crores of selling, these mammals, monkeys. And after the laws laid by the government, it has all come to very less. And now the experiments are already limited to small mammals, like what we have. So that's very important. And like ma'am said, we have got very advanced computational tools now available. Along with that, we have got stem cell technology, where you can almost create any human or animal organ. Once you can create human and animal organ, with the data available, you can create a digital model. That means you can create a digital heart, you can create a digital brain, you can create almost any organ, you can almost create theoretically, almost any mechanism. You can check the neurophysiological pathways, the effect of the drugs, it's all possible. But then this opens great opportunities for researchers like you to work. I'm very happy this topic is chosen. And my compliments to Dr. Anuradha and her entire team of researchers. We need to take a notch high. Like our Honorable Dr. Rahman said, she's got a great connect with the US researchers. We need to exploit that. We also need to explore industries like Fauna Analytics. Fauna Analytics is one big company which is dealing in this area. At the lower level, we have got MathWorks, which are dealing with a lot of computer simulation and virtual experimentation using biotechnology. On this day, Mr. Gaurav has arranged for a workshop, which has got biotechnology and life sciences component being taken on MATLAB. So my request is, let this be an extension. Let's study of virtual tools in biotechnology and taking them a notch better. For this kind of work, we can do a lot of simulation and a lot of research, our NTCCs in this very important area. So my big compliment to you, and let us continue having this workshop come international conference as a yearly topic. And it's a great topic. And next time when you have, maybe you can tie up with Springer to ensure that all the capable national and international participants, after a very strict peer review, it is loaded onto Springer and you can have due weightage and a larger number of highly acclaimed papers in Scopus indexed books. So that should be our aim. My good wishes and my compliments to all of you for taking on this conference. I know everybody is busy. We've got examinations, but nevertheless, it's a great exposure to all scholars in M-Pharm and to the youngsters in Pre-Pharma. Please make most of it, and to all the PhD scholars. Jai Hind.
Thank you, sir. Amity University always tries to appreciate the efforts of their collaborators and guests in all respect. In this regard, we are really privileged to share that we have a small token of gratitude to felicitate our speakers today. For this, Now may I invite the honorable dignitaries, that is Deputy Pro VC Sir, Dean Academic Sir, Kamar Rahman Ma'am, Director AIP Ma'am, our honorable speakers Dr. S.K. Raj Sir and Dr. S. Anbumani Sir and other esteemed dignitaries and our beloved organizing secretaries of AAE 2023 to join us onto the stage. I would now request our Honorable Deputy Pro VC and Dean Academic Sir to kindly felicitate Dr. S.K. Raj Sir with a memento and a planter, as we Emittans believe that giving plants to someone is to gift a new life in all. May I now request Director AIP Ma'am and Kamar Rahman Ma'am to kindly felicitate Dr. S. Anbumani Sir with a memento and planter. We would like to have few more minutes of you, sir and madam, as I am privileged to confer that this time AIP AUUP Lucknow campus is proud to inaugurate the abstract book of this international conference cum workshop AAE 2023 that includes abstracts of all accepted research and review articles for oral presentation competition. I request Tara and Amrit to present the abstract books to the dignitaries. I request Sir and Ma'am to kindly open these books, these abstract books on stage and inaugurate the same. May we have a huge round of applause for the abstract book, as it not only reflects comprehensively the paper's major findings in an easily understood manner, but also depicts the dedication, hard work of our scientific committee, organizing secretaries, and student volunteers, specifically others named from family third year. Thank you all. I would request you to kindly occupy your seats off stage. It's now time to get started with our technical sessions. For this, may I now request Dr. Lucy Mohapatra Ma'am, Organizing Secretary, AAE 2023, to introduce our honorable speakers to the gathering here and online. A warm good morning to all of you. Firstly, I extend my heartiest welcome to you all and all our enthusiastic attendees online to this inaugural ceremony of International Conference come workshop AAE 2023 organized by MIT Institute of Pharmacy, AUUP, Lucknow campus. Introducing a guest or a speaker is really an important responsibility that sets the stage for their presentation and creates an atmosphere of anticipation. I am privileged to have been provided with this opportunity to introduce our honorable speakers and guests of AAE 2023. Today, we have the privilege of being in the presence of a true visionary and researcher in the field of toxicology and experimental medicine. Dr. S. K. Rath is Honorable Speaker of Technical Session 1, Day 1 of AAE 2023. He is presently working as Chief Scientist Toxicology and Experimental Medicine, CSIR CDRI Lucknow. The Honorable Speaker portrays an astonishing research profile with over 83 articles published till date in several national and international journals of repute with high impact factors. He has over 4 books published to his credential and almost 7 different articles published in conference proceedings so far. The Honorable Speaker has got 1,360 citations and has H index 16. Further, Dr. Rath has been awarded with more than 12 awards by several esteemed organizations across the world. Some of the important awards includes Incentive Award for Technology Transfer by CDRI in 2021 and 2022, Incentive Award for International High Impact Publication by CDRI 2019. Besides, he has been associated with several recognized professional bodies as life member, such as the Society of Toxicology India. Today, his talk will be on alternative models in drug discovery and development. Sir, we are really privileged to have you with us today. Dr. S. Anbumani is the Honorable Speaker of Technical Session 2, Day 1 of AAE 2023. Dr. Anbumani has a keen research interest in deciphering the underlying cellular and molecular mechanisms of neurodevelopmental and complex behavioral toxicity by emerging environmental contaminants such as organic UV filters, microplastic, frame retardants, etc. in developing zebra-fish models. Presently, he is working as a principal scientist, React Division, Ecotoxicology, Laboratory CSIR IITR Lucknow. Ecotoxicology Laboratory at IITR CRK campus is the first and only government lab in India having GLP accreditation to conduct excitotoxicity studies. The Honorable Speaker has been serving as a regular reviewer in various peer-reviewed SCI journals and an expert committee member for setting up ecotoxicology lab in IPFT, Ministry of Chemicals and Fertilizers, Gurgaon, India. He has 5 funded research projects from several national funding agencies and 2 projects being funded by international funding agencies. Today, he will deliver his presentation on the topic zebrafish as a model to study emerging contaminants-induced developmental toxicity. In addition to that, we have Dr. Kavinda Hassan, Associate Professor of Human Anatomy, Histology and Embryology, College of Medicine, PNU, the Kingdom of Saudi Arabia as the international speaker to join us online today at 3 p.m. online. The esteemed speaker will make her deliberation on the topic that neural stem cell, a contemporary dual tool for disease modeling and therapeutics in regenerative neuroscience and an emerging alternative to animal models. Esteemed dignitaries and my dear attendees, I am also proud to announce that we have Dr. Singha Misra, Health Science Manager, Defense Health Agency, DOD, University of Utah, Germantown, Maryland, USA. Dr. Jasprit Singh, Scientist C, ICMR, National Institute of Pathology, New Delhi. Dr. Mukesh Nandev, Associate Dean, Research and Consultancy and Head, Department of Pharmacology and Pharmaceutical Biotechnology, DIPSA, University, New Delhi. Dr. Mohammad Ashraf, Associate Professor, Department of Zoology, AMU, Aligarh, Uttar Pradesh. and Dr. Sandeep Chaudhary, Associate Professor and HOD, Department of Medicinal Chemistry, OMC Lab, National Library, to join us as honorable speakers during day 2 and day 3 of AAE 2023. I'm glad to declare that the workshop on on training to handle computational tools and software will be decided by two eminent experts from our own department, that is AIP Lucknow campus, Dr. Rahul Kumar Maurya, Associate Professor, Pharmaceutical Chemistry, and Dr. Mohammad Yasir, Assistant Professor, Pharmacology, AIP Lucknow. Thank you all. I would now request Sriti and Vatsalya to proceed over this ceremony further. Thank you. Thank you, ma'am. It's my pleasure to invite Dr. S.K. Rath, sir, Chief Scientist, CSIR CDRI Lucknow, to present his talk on alternatives model in drug discovery and development.
Good morning, everybody. I'm grateful to the MIT University to invite me in such a wonderful occasion, because this subject of alternative models in drug discovery and development is one of the very, very intensified love in my heart. And I was the first person in CSIR to build a project on it in 2002, exactly 2 decades back, taking all the biological labs in CSIR into account. All 11 biological labs, they participated in this. One of them is certainly IITR, and all the CSR labs in Lucknow, they participated. And by that project, we actually achieved so many things that most of the labs, they started practicing in vitro experiments. Then, unlike the previous years where we did actually mostly rat and mouse experiments most of the time. So I am grateful to all of you for that reason. And I'm at the outset, I should thank Kamar Madam, who was actually our mentor personality in IITR. And just after the initiation of the project, Madam Kamar retired and we lost her from IITR, but she is still active. You see the energy she has in this age, which is actually very, very inspiring. I don't know, I can able to stand in her age in any platform. That is, Madam, you are really inspiration for us, and keep continuing blessing us and inspiring us. I'm grateful to Anil sir, who is knowing me as my involvement in MIT for last many years, because through J.K. Srivastav and Rajesh Tiwari sir, who, with his school I am associated for a long time, maybe 1 decade around. And many my students are also in engineering sections. They are working, for example, Dr. Yadav is one of my PhD co-supervisor. I was for her. So my colleague's wife also is there, with working in a different school here. So it's a homecoming type thing. Today's lecture will be actually a very basic, how this I will keep the correlation established between the alternative models and the drug discovery and development, because all of new drugs, right? Because the challenges are coming from cancer, especially cancer, and from pathogens, which are the drugs, whatever we have in our kitty, are actually not sufficing the purpose, because all of them actually develop, all the parasites and microbes, they have developed resistance, and we are struggling hard to getting cured out of those diseases because of the resistance. So new molecules are certainly required. So the question is how the molecules will be developed with different models and without spending much money in the animals, without killing a lot of animals. That is the question we actually address in drug research sector. and I'm sure Ambumani will throw light about the environmental toxicants, how they are putting lot of challenges and why and how the alternative models will help them. So the next slide is why we need alternatives first. This is the greatest question we have to ask ourselves. Each of you should ask the questions. Even if you are a common man, you should ask this question, why we need alternatives when we have all the things existing with us. As we require new models of cars, new styles of buildings, new universities, we also need new models to enrich ourselves to face the challenges. You have recently seen the COVID-19, how it has given the lot of challenges to us to find it out. But that challenge we actually faced very boldly. And India is one of the country which developed vaccines. Okay, one is totally by Bharat Biotech and ICMR. And this challenge we faced, and within 1 year we could able to develop the vaccine and actually start treating people. And besides that, not only in India, we actually served other countries' purpose by giving these vaccines to them. So this could be able to possible because we have lot of models there to test, because people immediately did all of knowledge, whatever COVID DNA knowledge or RNA knowledge was there. So we could able to make a vaccine which can be very fast done and can be checked actually in different models. And once we are found that it is very, very safe, then we can able to start. Okay. The one of the question Prime Minister asked actually, where the safety testing was done in Hyderabad in RCC lab, he asked one question: can you treat your Prime Minister with this vaccine? What is the confidence level? He told 100%. Immediately the next day, he flew to Delhi and he took the vaccine in All India Institute of Medical Science. So that is the challenge we faced. We confidently did it, and we could able to provide the all success, all the good stories, and could save actually a lot of people who are actually requiring the vaccine at this moment. So a lot of animals are being killed in experimentation, more than 100 millions actually, that is approximate number, which is used only in United States of America. In India, there is no calculation 1 decade back, but nowadays actually because of the Institutional Animal Ethics Committee, they set and CPCSA is monitoring them. So we have reduced numbers, but still we are also spending that much of animals every year, almost nearer to that in different private sectors and government sectors. Animal activities are proactive since 1920, and they are always trying to put everything in vitro. But I will have a question to all of you. Is it possible to do everything in vitro and without changing anything in animals? Nobody will actually come up with the idea that directly the human being can be tested with their new molecules or new vaccines, right? You must be knowing the Twitter war that all these vaccines, whatever India developed, are not up to the mark because they were not checked for safety. Lot of questions oppositions asked, lot of educated people asked this question, that how the government is pushing so fast to make the vaccine available for the people. But since the scientists are very, very confident, the government was confident, so government able to do it very confidently and provided it to the public. So always the people who are proactive, they will talk against them, against everything, whatever you develop. But that is a good thing, that you have to take the lessons from them, from the criticism they do. One has to take the lessons from them and go ahead for that. Then experiments of animals are not providing the desired results, which can be utilized immediately. That is very true. All the animals, millions of animals, we are dying, but we do not have a lot of molecules to get treated, right? The success rate actually out of 40,000 molecules, it is one molecule, it can come out as a drug. After that, if one, okay, thank you. So out of 40,000 molecules, one can actually get one drug. So see the 40,000 molecules you have to check preclinically in different animal models, disease models, then do the toxicity, then find out whether this molecule can be accessible to the humans or not. So naturally, the success rate is very poor nowadays because of the in vitro and other computational things. We are actually getting better things in our place, and the time of drug development is getting reduced from 20 years to 10 years. So that is certainly a good thing coming out of this, although we are actually killing a lot of animals, and desired results are coming only when we are using lot of batteries of test. Then resource crunch in maintaining large animal facility. For example, you have such a great university, but you do not have perhaps a animal house because it is very uh, cost intensive, and you require a lot of funds of different types and to maintain the animals. As Anil sir gave the example of the monkeys, the all over the country, one Sardarji used to supply the monkeys from Lucknow only. Unfortunately, in COVID-19, all his 3 generations are wiped out, and we have a new person who is supplying the animal named Tarjan Misra from Allahabad, and he supplies the monkey now. Very recently, CPCSA actually given the approval for supplying the monkey, and only 2 centers in today's day people are getting monkeys. One is Jaidas in Gujarat, Ahmedabad, and one is CDRI Lucknow, where a lot of monkeys are being utilized for toxicity and as well as for establishing the anti-malarial molecules. So if alternatives can give better advantage, then why not? Okay. So alternatives movement started actually from 1920s onwards. Slowly it has moved throughout the globe, and around a century, people actually traversed, and a lot of alternatives have been used for this drug discovery labs. So first, this 3R somebody told, Madam, the director of the institute actually spoke about 3Rs and few lines. This concept came actually from the principle of human experimental technique by WMS Russell and Bush. They actually described how you should handle the animals in a humanly way. Okay, when you are talking of killing animals, rat and mouse, in older days, for example, our supervisors' time, PhDs, most of the biological research only uses animals without doing any in vitro studies. So any molecule they get, they used to test in the animals directly and do the things. For example, there is a concept called LD50. LD50 is being utilized for both as development for the drug as well as the environmental toxicants. So in LD50 means out of 100 animals, how much concentration or dose of the compound or the new molecule actually required to kill the 50% of the animals. This is sometimes, if you think now, you will think it is very absurd. How a person can take 100 of animals and just try to observe the killings and death of the animals and go on counting and establish that this compound may be toxic or may not be toxic, or may be lethal or may not be lethal. So this is certainly a very disheartening research actually in those days people were doing. 2002 onwards, the LD50 actually we have blocked in this century. Okay, so around 80 years after the in vitro things, people started thinking that animal experiments for establishing LD50 should be blocked. So naturally, the alternatives are there, and I will be talking about while I will go through. So basically, it is replaced with other experimental models with accurate results. For example, a lot of computer simulations are coming in online. If you go and search many free softwares, you will find it out where you can find it out. Many commercialized things are there where drug discovery and development organizations, they are being utilized. For example, pharma companies, they are being utilizing. And they also try to replace with the lower animals, as Ambumani will talk about the fish, and a lot of Daphnia they use, a lot of Hydra they use to find out the toxicity of the environmental things. That can be also utilized for actually molecules which can be actually to be intended to be used as a drug. Then refine the experiment, which is reduce the number of the animals with meaningful results. That is strategies to get maximum data using fewer animals. For example, many parameters you check actually. One of the biologists, if you ask the questions, or pharmaceutics person, if you ask the question, people will tell that for different parameters, lot of animals they are being utilized. If you want to do that, you plan in such a experiment, you can increase 1 or 2 animals in the group, but you address all the problems like biochemical analysis like 13 parameters, hematology like 13 parameters, and reproductive organs also you can take and study the things properly, so that extra animals are being not utilized for that. So you have to reduce the numbers by planning well, and that is the best thing one can do actually, because without utilizing the animals, it is not actually possible, practically impossible to push a molecule into the market. Then refine the existing experiments so that less animals can be utilized, so that you can reduce the pain. Basically, all the things what you are talking of about killing the animals actually to reduce the pain, because you see the animal is actually crying. Those who are keeping pets in their house, they must be knowing how the feeling of the pet dog, they every day they interact, right? So similar pain must be happening to the animals who have a well-developed CNS system, central nervous system, so that we should avoid that pain and modify so that we can reduce the pain and distress to the animals. So the best adopted thing, which is nowadays in every lab you will find out, I would say with caution that this is not the ultimate. People are trying to develop maximum, but maximum knowledge we can gain out of the things, whatever possible way, so that we can reduce the animal. There exists a wide range of sophisticated computer models that simulate the human biology and the progression of the developing disease. But let me tell you that all these simulations are actually based upon our prior knowledge. Okay, if the prior knowledge is not there, your simulation everything will fail. Okay, so you have to know the targets, and if the drug disease target is not known to us, then actually it is not possible for any computer scientist to simulate and find it out whether the drug can be toxic or not, whether it can act in the target or not. So all these things has to be known prior to our things. So whatever molecule is there, we can give them a score of percentage like 90, 70, 80 or whatever it is, or not. Okay. So these models can accurately predict the way then new drugs will react in the human body and replace the use of animals. So one has to have the prior knowledge about that. Then only one can accurately predict the things. If the doctor doesn't know the disease activities of the diseased man, then he cannot actually prescribe a drug for the patient. So that is the thing why I am giving again and again the caution that one has to have a prior knowledge about the molecule, about the structure of the molecule, and about its physiological activity around the biology as well as in a disease condition. Then quantitative structural activity actually it's a very old thing actually in drug research. It has come actually in the very initially 3 decades, around 2.5 decades, and people are doing QSAR. Dr. A.K. Saxena, who was working in our chemistry division for a long time, for more than 30 years. He was actually a master in this city who does QSAR. Everything he knows about the QSAR, and he does a lot of things and developed a lot of molecules to in CVS area and CNS area. And you can, if you have get a chance, you can interact with them. We interact with him and find it out how the QSAR works. And I am sure you have a very good engineering set up here, and they can help you in using these QSR systems. Then laboratories are increasingly using QSR wherever all the I have seen like Cadila, Zydus, or CDRI, or Torrent was there, Ranbaxy was there. All of them actually using previously QSR. Nowadays also Cadila and Zydus, they are using this QSR. CDRI also does that. So what actually it helps in the initial test, it helps actually to the chemist to plan their molecules accordingly so that the interaction in the computation system will be more with the drug target, disease target, so that we can actually find out the better drug which can be further pursued to in vitro and in vivo systems. So this is a cutoff where alternative things can be utilized very efficiently to reduce the animal numbers in killing, because instead of going directly to the animals, you actually go to computation system, find it out the molecules. Once you are confident that 100% binding will be there through computer simulation, you actually pursue that molecule further for in vitro testing and further for in vivo testing. Then we have actually achieved a lot of goal. That is the various approaches we are utilizing all over the globe. People are working on test cells and tissues in test tubes or cell cultures. You must be knowing hundreds of cells actually are available. Our American type cell culture, one can buy from there and check from different cancers and for different types of things. You can also use, for example, in our CSIR, CDRI, we have for example, we have malaria. So malaria parasites are available here. Lot of African labs, they have malaria because malaria is actually endemic in those countries. So naturally, people have developed different types of malaria assay where you can directly test actually your molecule with malaria falciparum or malaria, because you get those malaria parasites and culture them in your lab, and you actually check whether this molecule which came out from the simulation actually working to kill the animal, kill the parasite or not. If it is killing, then only actually there is a logic that you can go ahead, because once you find that the malaria parasite is actually killing the animals, killing the parasites by the molecule, then only you can go further. Otherwise, it is useless to go further to kill the to pursue that molecule for as anti-malarial. So that is one of the good things. Anti-malarial, anti-filarial, all these parasites assays are being developed. JK sir is expert in that, and he has done a lot of contribution in those areas where parasites, anti-parasites can be developed. Then we have, as Anil sir told, a lot of mathematical people, they have a WhatsApp or something here, and I would request all the students to actually participate in such type of things, so that basic knowledge of how you can improve in developing a good molecule, discovering a good molecule for further studies in drug research can be done. And now, very recently, 3D tissue cultures in last 1 decade, people are doing from one cell to they make actually tissue, because a lot of criticism were coming that only single cell you are treating in different cultures, and you are opining that it is a anti-malarial or anti-filarial or anti-cancer or anti-liver toxic material. So you must actually work on a tissue system, and you must be knowing that when you are treating a cancer, sometimes even after operation, after the chemotherapy also, the patient revive again for a disease, right? For the same disease, relapses and goes back to the bad shape. So that is the reason why the 3D culture system is being utilized in this city. Dr. A.B. Pant in IITR, he does a lot of 3D tissue culture, especially in the neuronal cells. So if you are getting time, Madam Kamar is here, you can get a recommendation, and I am sure Pant will train you in how the 3D structures are being utilized. Then there is a organ on a chip concept that has come into actually picture a long time, about 1 decade. People are being utilized in United States of America. it substantiates the knowledge of our how the drug is being utilized and what is the target organ and what is the probable mechanism to work. But organ on a chip, people are not utilizing yet to utilize in drug discovery and development in this country. And if you have a chance to go to for a postdoc position in NIH, you can actually get the chance to work in this system and learn and come back to the country and employ it. Stem cell research, again, one of the major area. Even stem cell, now the recent guideline has come from drug CDSCO, that stem cell can be also utilized as a treatment parameter when the organ is being replaced with the stem cells, and people are trying to do it. And IIT Mumbai, and a lot of ICMR institutes, they are actually in a very, very advanced stage in this research. For the last 3 months, actually, there are 4 applications in DCGI where they are talking about stem cells and how they can be putting for stem cell therapy. So this is a good sign, actually, in this country slowly developing. And the non-invasive diagnostic imaging has actually contributed a lot. Okay, for example, you must be going for X-ray. Now it is ultrasound. Previously, people used to hesitate for X-ray test and other things because they are getting irradiated by the X-ray machine for a seconds. But ultrasound, people have acceptability, lot of acceptability. Even the child in your home also can be checked how the movement and other things, how the growth is there because of the ultrasound. Lot of imaging system has come now. Our institute has many animal imaging systems where we are actually in vivo imaging system. I myself handles all. We can see that throughout the animals, what is happening as the molecule goes inside and where it goes and seeds and how it is getting treated. So one can know that what is the target organ exactly, so that you can actually treat the patient very nicely. Then clinical research involving the human beings also people are utilizing, because all the knowledges in the clinical research, whatever a trial everywhere in the world is going on. So all the adverse reactions of the typical molecules are being recorded and they are being transmitted online to the different investigators to find out what are the side effects could be possible in this system. So this also helps in doing the things. To check the efficacy now, the animals used in drug discovery and development, lot of animals, as I must swear that we use lot of animals. But by the inception of the institutional animal ethics committees, we reduce the animal numbers, no doubt about that. You will be surprised that animal ethics and rights, other things people are talking in United States, but India is the first country to actually take the action from 1970 to actually in Parliament. Rajmata Gayatri Devi introduced the bill, private bill, to find out the animal ethics, and we have made a law in those days, but it was not implemented properly in this country. But in 2000 onwards, we started doing the things because of the pressure politically by Mrs. Maneka Gandhi, and of course, the organizations like PETA and other organizations, they forced the scientists to do the things properly. So that in 2005 onwards, we have ethics committees everywhere. So once the ethics committee is implemented, everything actually started regulated, so that the animal house cannot issue any animals without a particular IAC number or ethics committee clearance. Then the number of animals are actually controlled. Previously, people used to do that. If the 5 animals are dying, then the 6th animal they can get very easily. Is it not possible now? Now, to check the pharmacokinetics of the test item, we also use a lot of in vitro systems as well as animal systems. Now, lot of CYP inhibitions. For example, you must be knowing there are a lot of drug metabolizing enzymes in our liver, and all other organs have like kidney and intestines, they have some, and those enzymes actually breaks the big molecule into small molecules and get excreted. So that kinetics part of the drugs actually we all do nowadays in vitro system by using Kako cells, and we find out whether the drug can be inhibit the metabolizing enzyme or induce the metabolizing enzyme. That gives a lot of information so that the drug can be pursued further or not. Of course, the pharmacokinetics, how the body is managing the molecule in the body, has to be answered in the animals only. So that is the essential parameters we use actually in the rats or mouse or monkey to see actually in limited number of animals how the kinetics is going, if the things are going properly. Not only that, the kinetics also we check while studying the toxicity, so that we are sure that after a certain type of treatments, whether the molecule is going to stay in your body or totally getting cleared or not. So that is called as toxicokinetics. And we have that, although we have half of the things we replaced, like for example, Pampa, we use, that is again the solubility and from one membrane to the other membrane based on the epithelial cells, how it is crossing the borders and epithelia passing the borders from gastrointestine to the blood vessels. All these Pampa tests actually say us, and we do that regularly to do that. And pharmacodynamics again is a challenge where essential animals are being utilized, so that minimum number of animals can be utilized without giving, because we have all prior knowledges. So limited number of animals you use and reduce the all alternative things. So or reduce the animal numbers by using the alternative things. Then pharmacology again is a problem. The part of pharmacology one has to, because after taking, for example, alcohol, the behavior only can be tested after taking the alcohol, right? You cannot find out the behavior without in alternative model. So they are the part where essentiality is required, and the pharmacology also we do minimum number of animals to do the things. Safety is the priority for any drug researchers. So safety, in most of the safety studies, we use the animals even today, but we have reduced because we have made a compact system so that we can do the. For example, previously people used to do a lot of animals in repeat dose toxicity studies. Hundreds of animals are being utilized in different conditions. Actually, nowadays we and the drug regulators also trying the same thing. The treatment has to continue till the indented doses and durations in humans has to be defined properly. Then only the treatment can start. For example, if you are treating the humans for 15 days, so 15 days treatment only has to be done. Previously, it was actually a lot of days, around 100 days, 300 days, 400 days. Now it has reduced to actually the fixed days. For example, 15 days means 15 days treatment in the animals. 30 days means 30 days treatment of the animals. And beyond 90 days, no drug research. Actually, beyond 90 days, if it is finding safety, it is fine for us, and the regulators accept that. For Ayurvedic products and plant products, perhaps they require 180 days, but we are only, majority of the drugs actually we tested for 90 days. And we have a recovery period also testing, so that another 7 or 8 animals you take, so that we are confident that the whatever problems we are getting in the highest dose, whether it is getting recovered or not. In that way, we actually reduce the animals in the system. So mostly the mutagenicity systems are actually in vitro in nature, and genotoxicity also in vitro nature. But reported toxicity, you cannot actually made a replication in the in vitro system or alternative model. So we have been utilizing this, and systemic toxicity also. We can check biochemical parameters in vitro, but it exactly doesn't give actually the what it can make in the living system. So that is why where the compulsion is there, so we have to check in the animal models. So some of the successful models we have, for example, microbes, and that is mostly basically actually Salmonella and E. coli. Yeast, we have very good system to study the mechanisms. Microbes like E. coli also helps us to make different types of knockouts and transgenics, and helps us give the information about the mechanism of the molecule. C. elegans, again, a very, very transparent system where you can see actually 900 plus cells, and each cell you can observe under a microscope, and you can see what are the inside going on, okay, on the microscope, so that you are very, very confident that the molecule can bring some effects or not. Lot of C. elegans mutants are available in this city. I think in CMAP people are working, in IITR people are working, in CDRI we have 60 type of mutants of C. elegans, and they are being utilized in different purposes to find out the mechanism of the action of the small molecules. Then we have zebrafish. I think the Ambumani will talk about that. This is a beautiful system because a small fishes you can grow in the aquarium, and you can see through model actually for anti any blood pressure and cardiac toxicity. If you want to find it out and how it is affecting the cardiac system, this is the best model. You can see through and find out what would be the fate of the molecules. Then organoids, as I have told, people are very, very successful in neuronal organoids. Lot of labs in the United States of America and EU, they are using, especially in John Hopkins, one madam who is a director, she talks about all the organoids, how she can able to use it properly to evaluate the toxicants. Then organ on a chip already I have spoke about. So at the design system, I have already told that computer aided drug design system. But once you have taken a system, you please do not buy anything commercially until unless you are experienced properly. Lot of free ways you are finding out in the Google. You can Google and get it out. For example, in recent last week, actually we rejected a computer system, computational based system, where actually only 30% success was there. So the success is actually not more than 80 to 85%. But if you are using a 30% success thing, then it is actually maybe misleading things. As I have already told, previous knowledge is being introduced in this simulation system, so that whatever is known should at least can be validated here. So while validating for 1 month, we could find out that only 30% of the molecules actually this system is identifying and others are not. So that is a very disastrous things for the researchers. So one has to be very, very careful about while using the system. Disease models, a lot of disease models, in vitro variety cells and parasites, transgenics are being utilized and being commercially available. You please, wherever you are trying to do small researches, you start with in vitro systems, in vitro cells, and go on doing the things. The target is to reduce the disease phenotype in a required time period. So wherever the model have a disease phenotype, you get it and get implemented in your system, and then actually validated in the in vivo system, and you can do that. For example, in malaria, I told in different malaria parasites, you do in vitro, and then you tested actually in the lot of mouse, for example, 5 or 10. Then again, go back to monkey models and do the things. All the parasites will not work as in human. So you have to be very, very careful. All these SIP testing, as I have told, all 8 molecules, they actually all SIP 1AB, 2B, C2C, all these are drug metabolizing systems, and the DCGI or FDA, they require it 8 type of SIPs, how it is working. So everything can be in vitro now, and people are doing in any labs, and you can also try to do the things in your lab very, very efficiently. And Pampa, as I have already told, it's in permeability, and one can do it very easily, so that permeability can be tested. Pharmacodynamics, we do it in vitro models to test the item successfully, especially very in antimicrobial, antiparasites. In other cell types, actually, it is very, very difficult. Cancer cells, of course, we are doing, but it's not very, very accurate. So I will caution those who are doing the things and pharmacodynamics using the in vitro cells. pharmacology, I already told the neural studies are very good, successful regeneration, and in advanced stages of molecules are being in clinical trials also after getting tested in these organoids. The major challenges actually now is how far we have actually got the success in toxicology studies, because people have an eye always in toxicity studies. How people are telling us, public is telling us that you are killing a lot of animals in the name of safety evaluation. But let me tell this: if you go to this table, then you can see that mutagenicity studies are totally replaced. Okay, in mice previously we used to do, it is replaced by the Salmonella typhimurium, which is developed by Bruce Ames in 1972 by Bruce Ames. Now it is actually in every lab throughout the globe. Wherever mutagenicity is being checked, they have Salmonella and E. coli, and they do that. Then micronucleus assay previously we used to do in mice, and now no more mice are being utilized, and we are doing in the cells, and we can able to do that. Chromosomal aberration assays, which is very important assay, and that is actually again in vitro we are doing in human cells, so that human relation also we can establish by using this. LD50 we have replaced that with maximum tolerated dose by checking 3 doses, 6 animals, and you can find it out whether the maximum tolerated dose is within the 2 gram or not, because all over the globe people are actually trying MTD has to be established maximum 2 gram. Beyond that, if it is there, actually it is the molecule cannot be utilized as a drug. So that is the limit we have set. So automatically the animal numbers reduced in that. Then repeat dose studies, as I have already informed you, that only the intended doses and duration are being utilized to plan the toxicity studies, so that it can be automated analyzed systems with accuracy. And after that, the repeat dose studies are being tested, started, then toxicokinetics in addition to that in the same animals, so that we can reduce the animal. Of course, there is fertility studies which cannot be done in vitro. We have to go for fertility and other things. Whenever it is the drug is required to be tested in the females, certainly we have to check with fertility and teratology, and this is an essential component. Many a company, they used to tell that we will not use it in females, but automatically you do not know that whether after the marketing, the molecule can be tested in the females. So therefore, always in DCGI, I used to thrust upon that it has to be compulsion that all the test items should be tested for female fertility and teratology. So with this, I think I have finished the talk in time, and any question you have, please do ask me so that I can able to answer you if I know the answer. Thank you.
Thank you, sir, for enlightening us with your nice and elaborate talk. Now the forum is open for the question and answer session. If there are any questions or queries from the audience, online or offline, we would love to take up. We will take few questions, and with due permission from the honorable speaker, sir, we will share his mail ID to our honorable attendee. Yes, sir. His mail ID is also been provided in the slide, the first slide. So if ever we are unable to take questions due to constraint of time, so we will forward the queries to the speaker to be addressed later. Any queries right now? If anyone? So then in that case, we'll forward you the queries online. Thank you, sir, for this elaborative talk. If we are going to take the principal over the meeting. Thank you, ma'am. It's my pleasure to invite Dr. S. Anbumani, Principal Scientist, React Division, Ecotoxicology Laboratory, CSIR, IITR, Lucknow, talk to talk on zebrafish as a model to study emerging contaminants induced development toxicity. So please. Sir, we have a query from our online, one of our online activities. Meanwhile, I request sir to carry on with the presentation. Sir, we have a query from one of our online activity, and he's asking that what are the important main limitations for alternative to animal experimentation in health system?
As I understood, the question is, what are the limitations, right? For alternative in drug discovery and development, right? Yeah, the first limitation is the animal is not the human, right? So whatever you do in alternatives, people will accept only where it is logical to them. If the same thing is working in human, or it is seen in animals, then only it is working. For example, dog is a very good model for CDA studies. Okay, because there is lot of similarity. When there is dissimilarities, for example, Hydra people are utilizing for identifying toxicity. Fish people are utilizing for identifying toxicity. But people will tell this is a fish and we are humans, we cannot be accepting this, right? None of us will, right? As the COVID-19 vaccine people are challenging, similarly safety, when perception is there, unsafe is there, unsafe, similar molecules can be toxic if you modify methyl or ethyl or nitrogen group, you have toxicity, lot of toxicity can be.
But
Okay.
I would just request you if you can join for another 5 minutes on the stage because we are not audible and visible to the online participants. I do hope that answer will be dispatched and long. I will just take another
Hello.
query.
Hello.
Yes, sir.
Okay.
Now you're audible
So-
and visible online, sir. So the next query is which type of study is better to evaluate metastasis in vivo or in vitro way?
Metastasis actually is a special phenomena, right? Because cell to cell contact inhibition, right? That stops. These cells try to move within the blood cells and goes from organ to organ, right? So that similar things are not happening anywhere in the other system. So metastasis can be only studied by making a cell which is actually made up of all cells. That can be done today in vitro, and then study how this is handling the things. Okay, so you have to disturb the genetics of the molecules or genetics of the cell, then do the things. Most of the cancer cells actually, whatever we are growing in the lab are actually cancer cells. So most of the metastatic questions are being answered by using HeLa cells or breast cancer cells like MCF or triple negative cells, MDM2 cells. All these cells are actually cancer cells and they are in metastatic in nature. So there is no problem. If you want to ask some particular question in metastasis, then you have to actually modify that cell in the genomic level and do something else by transgenic or knockout things and get this knowledge out of treating that cells and do the things. Any more question? Done?
Yes, thank you.
I hope I answered your question.
Sir, thank you. Thank you for addressing the question, and now I would request SR Minister to continue with his talk.
A very good afternoon to everyone. Today, my topic is covering three major aspects. One is, as what Dr. Professor S.K. Raj said, replacement with lower animals. So I'm going to talk about this lower animal, which is a tiny fish with a big splash in the area of, you can say, both the discovery and the toxicology. That is none other than the zebra fish. And the second thing is application of this model to study the mechanism of action of different xenobiotics and the usage of the computational tools. How can we complement each other? That is the findings from the research experiments and also the observations we got it from using some of the molecular docking tools, how they both are complementing each other so that we can still get the robustness and integrity on the result we obtained from our experiments. So before going to this talk, I would like to represent my organizations, where I am and what I am. So CSIR IHE, or Indian Institute of Toxicology Research, the motto is safety to environment and health, and obviously servicing to the industry sector. And the vision is from laboratory to the field for societal benefit by contributing in the niche areas of toxicology, the different domains. And the mission is to be a world leader in the toxicology research and endeavor to mitigate problems of human health and the environment. So coming to my lab, so I am associated in the regulatory toxicology. The name implies regulation of the chemicals usage and the safety of the chemicals in order to get the product testing and its public usage. So particularly from my lab, ecotoxicology laboratory, we are studying the ecotoxicity of emerging contaminants on aquatic and terrestrial organisms by the acronym ECATO. So under this thing, my laboratory is studying on different tasks such as utility of this small animal to assess the developmental neurotoxicity. And also we can use this particular model to generate some disease-based models, particularly related to the behavioral diseases such as autism spectrum disorder. Apart from that, we are also working on different models such as earthworms to assess the risk associated with the land or the soil. And also we have other smaller animals such as Daphnia and the algae to assess the impacts on the freshwater bodies which are affecting our trophic chain. So the ultimate goal of doing this multiple task is to create an alternate to the animal model for effective chemical risk assessment and also for an effective ecological risk assessment of these two ecosystems of aqueous and the terrestrial. So these are the available model systems in my laboratory, starting from algae, Daphnia, and zebrafish, which covering a basic trophic chain which we studied in our school books, and the zebrafish, and we are using their embryos also. I'm coming to the terrestrial models. We are having earthworms, Japanese quail, and the honeybees also. So this is safety. The motto, as I mentioned, safety to environment and health and knowledge creation to that ecotoxicological databases for a number of chemicals by conducting the safety evaluation studies so that we can service the industry also and academia also. And obviously will become the leadership in the regulatory framework. So let me come into the topic. So today I'm going to talk about one of the emerging environmental contaminant, organic UV filters, which everybody knows this is one of the chemical ingredients widely used in sunscreen products and shampoos and also textiles and coatings in order to prevent the degradation caused by the UV radiation. In terms of cosmetics, it is used to protect the skin from the UV radiation to prevent the deleterious effects on our skin causing melanoma or the skin burn. So you may ask the one question, since they are using in the cosmetics, why we need to bother about the environment, how it will come to the environment, what will happen to the environment once these chemical entities will come. Obviously, they will come to the environment because of the wide usage by us, by humans, and also enormous production. So ultimately the end disposal is at the environment, either through the wastewater treatment plants or through the sludge from the wastewater treatment plants. By these activities, it obviously reach both the water systems and also the soil matrices, and the biota that is called as the non-target organisms will get eventually affected. And that is the reason why we want to study what kind of adverse effects this include on these organisms by using this particular model, the zebrafish. So these are some of the cosmetic products containing this particular organic UV filter, which I'm going to talk today is nothing but a camphor derivative, 4-methyl-benzylidene camphor, which is widely used sunscreen product in lot of the cosmetics by the name enzacamine at different concentrations from 4 to 10% as recommended by the Bureau of Indian Standards. So as I mentioned here, they reach this environment through personal care products and also is used in plastic items and also it is used in food packaging materials. So to prevent the degradation by the UV radiation. So ultimately it will end disposal will be in the water bodies and the terrestrial ecosystems. Thereby it is causing detrimental effects on these non-target organisms. Besides that, because of this external emerging, enormous use, we have find out from different matrices. It is getting accumulated in the different matrices because of their high lipophilicity on freshwater, in PPB ranges, and river and sediment, and including in humans' urine and plasma also, because of the extensive use in nanogram per ml. So obviously, the non-target exposure is highly inevitable. So we need a environmental toxicity is a major concern with this kind of chemicals. So these are the currently available information regarding these cosmetic products in this different model systems from primary producers, consumers, and the secondary consumers. So they affects the growth, they will inhibit the reproduction, or it may causes infertility in the exposed animals and all those things. And it also leading to the oxidative stress and endocrine disruption. But still there is an unanswered question, that is, what will happen when the development, what will happen to the vertebrate development effects when such kind of exposure occurs. So this particular vertebrate developmental impact using this particular model organisms, we studied using this tiny fish in our laboratory, and we got some interesting findings also. So why zebrafish? What is the need? Why we want to use this zebrafish? As Professor Rath explained, we can use the replacement using the lower animals, which is suffice the 3R strategy: reduction, refinement, and the replacement. So the one major advantage of why zebrafish, it is a OECD recommended model systems. OECD is nothing but an organization for economic cooperation and development, which governs the regulation of the chemical usage. So they have set of guidelines to conduct the toxicological and the environmental toxicity studies, like how we are having CDS-CO for drug-based things, ICH guidelines and all those things. They have their own unique set of guidelines to carry out the toxicity studies. So they recommended this zebrafish as one of the model system. So no one will stop you to work on these model systems. Nobody will ask you the question why you want to use the zebrafish for your research, even though you need the ethical approval for this model, because obviously you have to get the ethical approval through your IAAC, because this also feels pain and the distress while handling. So any organism which experience the pain or well-developed nervous systems, they undergo the ethical approval. And second thing, it is easy to maintain. Unlike the higher animal models such as rat, mice, which Dr. Rath said you need an extensive animal host facility. This you can maintain this from small aquariums to a big standalone system, so that the maintenance cost is very less when compared to the higher animal models. And the second advantage is it gives a high clutch size. From a single pair of zebrafish, you can get 300 to 400 embryos for your experiment, so that you can conduct on the same day, you can conduct as many number of replicates and as many number of times you can repeat your experiments for your data integrity. And second thing is it has a developmental transparency. The embryos of the zebrafish are developed is external. So you can usually visualize the blood flow, heartbeat, the what you call it is the cells developing, the organs that are getting developed, you can able to see under the microscope, so that it will be a see-through model can be able to achieve with this particular model system. And another important thing in the drug discovery and the pharmacological aspects is the HTS, nothing but the high throughput screening. At a time, you can screen n number of molecules, lead molecules, without going into the rat or the mice model. So this can be used as a screening platform for your lead molecules for particular specific activity, which give you a result, so that you can carry your next preclinical trials on other animal models. And other major advantage is that its whole genome is sequenced of this particular model system, so that you can able to play at the genomics level also, so that you can able to knock out some of the genes of your particular disease target, and you can study the signaling pathways. And obviously this many advantages makes this as a suitable alternate to the higher animal models in the area of pharmacology and toxicology research. So we ask one of the key question, when this 4-methyl benzylidene camphor extensive usage when it comes to the environment, does it have any adverse impact on the zebrafish development, behavior, or reproduction? Does it have any impact? So this is our key research question. For this key research question, we use this approach called the cradle to the grave approach. That is from the birth to the death approach we used it. This is one of the classical approach we widely used for chemical risk assessment and also in the drug discovery aspect also, cradle to the grave approach, so that we focus on three major verticals. One is the development, and one is on the neurobehavioral aspect, and one is on reproduction toxicity and the progeny health also. So all these three things can be achieved using this particular simplified model. So this cradle to grave approach we use to address this question. So for this developmental toxicity, we just determined the lethal concentration of this 4-methyl benzylene camphor on zebrafish embryos, and also studied some of the developmental endpoints at different biological levels of organization, starting from cellular level to the molecular level, to derive the mechanism of action of this chemical in this zebrafish. So this is our experimental plan, how we used it. We used to maintain the zebrafish in the standalone system in our laboratory. And then we used to get eggs from 2 male and 1 female ratio. And the fertilized eggs will be were exposed to different concentrations based on the OECD guideline, fish embryo acute toxicity test to determine some of the endpoints such as embryo coagulation, heartbeat, detachment of tail, and the lack of somate formations in order to derive the LC50. Like Professor Rath said, LD50 in terms of mammals. However, in case of zebrafish and other smaller animals, aquatic organisms, we used to call it as LC50, that is lethal concentration. The reason is we are not injecting the animals directly based on the body weight, because these are all in the media. So we used to expose the xenobiotic in the water. That's why it is called as the lethal concentrations, LC50. So when we exposed this thing, it is only a four-day experiment. This is a 96-hour. Within 96 hours, you will get the LC50 value of any compound. This is one of the main advantage of using the acute toxicity. So if you look at these things, this is a control one, which is a well-developed embryos with a tail, everything is detached. However, this is the coagulated embryo. This is no longer viable. And this is something that tail detachment is not happening. And this is unviable embryos. So these are the four major endpoints. Because lack of heartbeat, you cannot show it as an image. It will be visualized in the form of a video file. And we exposed it to two different exposures. One is continuous exposure, and another is every 24 hours we renewed the system. Because exposure, chemical exposure or any drug exposure, you want to ensure how much percent the organism is exposed. That is more important. At least for an toxicology or in the pharmacology, 80% of exposure is essential to correlate your findings to the toxicity. So in order to find out that 80% exposure is there, we every 24 hours we used to renew the media based on our bioanalytical research. So we subjected our media to UHPLC analysis to find out how much percent of this particular compound is present in that particular media. Accordingly, we will devise the time of exposure. So based on the exposure pattern, the values of the LC50 also will be changed. So then based on the LC50 value, we chosen some sublethal concentrations or otherwise the environmental concentrations, because our aim is to see the developmental effects, neurobehavioral effects, and the reproduction effects. All these three things will happen only at the lower doses, because at higher doses, everybody know it causes the mortality or the lethality of the animal. So we need to expose them at the lower doses. So we chosen three major concentrations, 550 and the 100. 1000 we excluded because 1000 also we have seen some larval mortality. So this is the graph showing the how much concentration of the compound is remain in that particular media. So by 24 hours, 50% of the compound get degraded in the media. So that's why we have chosen every 24 hours we renewed the zebrafish embryos with this particular model compound. Then we conducted up to 10 days, we continuously exposed the larvae. So from the embryo, we now look into the effects on the larvae also. So we identify the larval LC50 also at around 500 ppb of this compound. So based on this, we devised 10 times lower than and 100 times lower than of this larval 50 to study the neurobehavioral, developmental, and reproductive effects. So this is our work plan, schematic work plan for our biochemical effects of this particular thing. We used some of the marker assays such as multi-xenobiotic resistance, oxidative stress, oxidative tissue damage, apoptosis, and the apoptotic pathway using the transcription and translational gene markers. So when we exposed this, our embryos, fertilized embryos to this model compound at 3 different concentrations, we noticed a significant reduction in the heart rate. So it is having some effects on its cardiotoxicity, it induces, and also it delays the hatching rate. It affects the hatching potential of the zebrafish embryo, and classically it induces a developmental deformity called axial curvatures affecting the trunk that is called as the scoliosis or axial curvature, which is a characteristic symptom of this particular model compound 4-methylbenzylidene camphor. And because of this, the larval length is also significantly reduced at the 50 and the 500 ppb of this compound. So this indicates this particular compound affects the developmental endpoints in this particular model system. So in case, so then we thought this is a panel image showing these are the different stages of the development of the zebrafish embryos from 24-hour to the 120 HPF. This is the control one. So if you see in the 72-hour, by third day, the controlled embryos will use to hatch out in the form of a larvae. But however, if you see at the exposed ones, still they are not yet hatched. So the hatching is an important thing in the development of the zebrafish. So this particular compound inhibit the yolk gland and the hatching process of this zebrafish. So then next we thought the question. So this induces the developmental toxicity. How then it will reach us into the zebrafish embryos? So we carried out a multi-xenobiotic resistance activity. So we using a pharmacological inhibitor, the cyclosporine, as a positive control A, which inhibit the ABCB4 glycoproteins on the cell surface. So this is one of the marker used for the drug also multi-xenobiotic resistance activity. So when we carried out that, then we saw the accumulation of rhodamine in the treated ones. So this accumulation of the rhodamine shows that MXR activity is inhibited by the 4-methylbenzylidene camphor, which allows the toxicants to enter into the cells of the embryo. And we also confirmed this, our multi-xenobiotic resistance activity results with molecular docking of the ABCB4 protein using the molecular study, which we find 4-MBC has higher affinity to this particular ABCB4 modeled protein with a binding energy of minus 8.5 kilocalories per mole. So this clearly shows that this 4-MBC has the potential to enter into the zebrafish cells and it causing the developmental defects. Okay, next, it causes the multi-xenobiotic resistance activity. Then obviously it induces a stress response. What will happen to the stress enzyme parameters? Whether based on the stress enzyme parameters, whether it causes any tissue damage at the oxidative tissue damage or not. Then we noticed the expression profile of the major oxidative stress enzymes such as SOD, CAT, GST, and MDA, which gives a temporal and biphasic response between 5 dpf and 10 dpf, and the levels are increased at the higher dosage, indicating the organism is under oxidative stress upon exposure to this. This particular oxidative stress eventually evident at the tissue level also. We carried out using the XGAL activity, that is the SA beta galactosidase activity, which gives a blue color form after interacting with the compound 5,5 prime dibromo 4,4 prime dichloro indigo, which gives you that presence of the oxidative tissue damage in the presence of 4-MBC, which indicating the presence of this indigo blue color. Wherever in control you are not able to see the intensity. So based on the fluorescent image, based on the software, we used to derive the positive pixels per larvae, and we noticed that there is a significant enhancement in the formation of the blue color in the treated ones compared to the untreated ones, indicating the significance of oxidative tissue damage because of the effect of 4-MBC affecting the oxidative stress markers at the cellular level. Okay, this oxidative stress, now we find out that both the tissue level and the cellular level. So do they have any impact on the molecular level? That is next our question comes into our mind. So now next we carried out apoptosis. We studied using the acridine orange assay, quantitative acridine orange assay, and we noticed the extensive accumulation of the acridine orange in the highly 50 and 500 treated animals, indicating the fluorescence intensity, showing a statistically significant enhancement of the formation of dead cells in the apoptosis and the zebrafish. So considering this, now we target some of the prominent markers of the apoptosis, such as the caspase-3, BCL2, and the BAX genes at both transcriptional and translational level. And we noticed a significant increased expression of these markers, caspase-3, BAX, and the BCL2. But however, BCL2 is getting decreased because we all know BCL2 is an anti-apoptotic protein. And also at the same time, if the apoptosis is more, it won't allow the cells to evade the protect the apoptosis. It will allow, instead it will allow the cells to undergo the apoptosis. That is the reason why we get a decreased expression of this BCL2, because BCL2 is also called as the lots of the death also. Besides, it is having this preventing potential of the apoptotic. And this is further validated with the translation studies using our Western blot analysis. And we found the increased expression of the BAX, caspase, and the BCL2 also. So at the end, we can say that this 4-MBC induced the developmental toxicity by inducing apoptosis and the developmental deformities through the apoptosis and triggering the oxidative stress at both cellular and the molecular level. So next we thought that, okay, if this is inducing the developmental toxicity, during the developmental toxicity, we saw the larvae are not moved when compared to the control group. So we thought there might be something related with the nervous system getting impaired with this particular zebrafish upon exposure to the 4-MBC. So now we look into what will happen to the effects on the nervous systems, because we didn't find the exposed larvae are not moved upon the response. So we carried out some simple behavioral responses such as locomotor activity, thigmotaxis, and the touch response. These are some of the basic behavioral endpoints. You can use it for both chemicals and the drugs that have impact on the behavior, that is including the movement, or the thigmotaxis is nothing but the wall hanging behavior, which is an indicator of an anxiety phenomenon. So what we did after exposure to the 4-MBC, we subjected our animals to these touch responses. Parallelly, we also see the histopathological effects on these particular model systems. Because since this is an effects on the nervous system, we cover both central nervous system also and the peripheral nervous system also, because the movement is associated with the coordination of the peripheral nervous system from the trigeminal sensory neurons. So surprisingly, we noticed the percent thigmotaxis, that is the number of larvae that has go and adhere to the wall of the Petri plate. Usually it is, if they are normal, they will remain in the center of the arena. Instead of center of the arena, they, because of the anxiety, they will go and stick towards the sides of the Petri dish. So that is called as the enhanced thigmotaxis. So this is significantly increased when we expose to the 4-MBC. And also we did a touch response. This is a simple response. We have just taken a small pipette tip and touch each and every larvae at the head region and the tail region, and we will wait for 5 to 10 seconds to see its movement. But the movement is drastically reduced when we expose things. So then we thought that if the movement is getting affected, obviously there is the effects on the peripheral nervous system, which will have impact on the central nervous system also. So this is further again clearly shown from the microtracker based assay, which is ARENA, which induced the significant movement reduction of this particular larvae upon exposure to this compound, which also have, we note a positive correlation between the length and also the movement of the larvae. Finally, we also noticed a significant increase in the ACHE activity, acetylcholinesterase and esterase enzyme activity, which is a classical marker for the neurotoxicity, also getting affected upon exposure to this compound. So now we thought, okay, let us go and see what we notice the effects on behavior, whether this behavior is associated with the central nervous system or not. So we did the histopathology of the zebrafish. So if you see, this is our control animals, and this is the forebrain, and this is the midbrain, and this is the hindbrain. If you see at a different objective, if you see at the 20x and the 40x, the neuron cells in the brains are populated in the control. But however, when you see the exposed ones, the cell population is getting decreased. So this is clearly evident because of the decreased cell population. They are not able to send the coordination for the responsible for the movement in the larvae, which is shown the reduced movement in the behavioral assay. And this is on the 10-dpf also we have noticed. However, there is a slight improvement in the 10-dpf exposed animals, where some of this, because this is a growing animal. So obviously once the cells were removed, obviously new cells will be, progenitor neuron cells will get replenished in the brain. But still the effects are persistent till 10-dpf. And we also noticed particularly this portion is the Purkinje cell layer. So everybody know Purkinje cells important function is in the coordination of the movement. So then we thought, okay, this is having some of the effects on the neural progenitor cells. So then what will happen to the genes and the proteins that are encoding the neural progenitor cells? So we chosen some of the important genes which are mainly involved in the zebrafish neurogenesis, such as SHA, sonic hedgehog, neurogenin 1, ELBAL13 alpha tubulin, then alpha 1 tubulin, and MBP, that is the myelin basic protein, which covers the myelin sheath of the neuron. So these all genes, when we did the qRT-PCR analysis, we have seen they all get the increased expression profiles when compared to the untreated ones. And particularly, we have noticed on both 5-DPF and the 10-DPF also of both the marker set of genes. Then we further out downstream our experiments to at the translational level. At the translational level, we have focused on the SHH only. Why only particular SHH? You may ask you may ask one question. There are so many genes I have studied at the qRT-PCR level. Why have I chosen only the SHH at the translational level? So this is I would like to say a basic thing. This is our mammalian trigeminal neuron that is getting specificated in our brain development. This is the zebrafish trigeminal neuron, which we call it as the Rohan-Behead trigeminal neuron specification in the brain. So these Rohan-Behead have the dorsal root ganglion, Rohan-Behead, and trigeminal. All these three neurons' main function is in the sensory nervous system, triggered by the central nervous system. How it get triggered? That is through the sonic hedgehog signaling pathway, because the sonic hedgehog signaling pathway is one of the important critical pathway in the any organism's development. Because this particularly in zebrafish embryogenesis and development, this sonic hedgehog signaling directly regulate the expression of neurogenin. So this is the results show that SHH has directly inducting the neurogenin 1, and this neurogenin will again triggers the trigeminal neurons, which is responsible for your touch, pain, and temperature sense. So we can clearly see the neurogenin expression levels were increased after exposure because of the impairment in the sonic hedgehog signaling pathway, which affects the movement of the larvae, which we clearly seen in the behavioral studies also. Now it is molecular level also, we have confirmed that yes, this neurogenin levels were enhanced at the translational level, which is highly significant, which causes the movement impairment in the zebrafish model affecting the sonic hedgehog signaling pathway. So this clearly shows that apart from the developmental effects induced by this compound, it affects the movement of the animal by affecting on the peripheral sensory nervous system by causing the motor neuron deficit. Okay, now the next question we ask is, okay, now it causes the nervous system also. What will happen when we leave these animals till when they reach the adult stage? How? When they will keep the adult stage, and then we will see the reproduction, because ultimately reproduction is the ultimate outcome of any animal to enhance its lineage in that particular environment wherever it is residing. So next we thought into reproduction. Before that, I would like to show some of the molecular results also. Like SHH, we studied because of its direct involvement in the neurogenin. We modeled this SHH because direct zebrafish SHH morphogen is not available. So what we did, we take the human SHH and we modeled it using the PubMed and the PyMol software with the computational toxicology group from our institute, Dr. Parthasarathi. And that group, they did the molecular docking and the molecular simulation studies and noticed a strong binding affinity of this 4-MBC to the SHH protein. So this is also, we can now prove it in computational also, we can prove this 4-MBC has significant affinity towards the SHH protein, and this is we cross-verified from our experimental approach also. So now both are complementing each other. So this is the mechanism we can say. Upon exposure, the larvae reduce the compromised their movement based on the touch and the response by affecting the sonic hedgehog and the NGN upregulation, causing the damaged neurons in the peripheral sensory nervous system by affecting the synaptogenesis, which prevent the synapse formation. It's not unable to send the signals from one clastic cleft to the other clastic cleft, so that the movement is getting impaired. So this is the salient findings. We noted the sonic hedgehog and the neurogenin was affected at transcription and translational level. At the histological level, we can see the brain cell populations were reduced. And until date in the literature, particularly for this organic UV filters, there is no information about the effects on the peripheral nervous system. And our report is the first report on the 4-MBC that is affecting on the peripheral sensory system in zebrafish developing stages. So now the next question comes, as I mentioned, okay, now at the development stage, early stages, this is affecting the development of nervous system. What will happen at the later stages? Because early exposure of chemicals or drugs may not induce effects that immediately, it may induce the detrimental effects upon the later stages, that is when the organism reaches the adult stage. This is also applicable to us also, human beings also. So what we did, we gave the treatment up to the embryonic larval stages only. Only 4 days treatment. After 4 days, we maintained these animals in the chemical-free medium till 180 days. For 6 days, for 6 months, we maintained them to grow into the adult stage. And in that adult stage, we assessed the how much number of animals were survived, their body weight, their clutch size, their fertilization rate, and their effects on the F1 progeny also. Besides, we also studied the effects on the endocrine disrupting molecules such as estrogen receptor and the androgen receptors and the vitellogeny, which are using both in vivo and the in vitro. So when we see the, based on the computational study, we have noticed this 4-MBC is having affinity towards estrogen receptor and androgen receptor. So this affinity of both estrogen and androgen receptor clearly says that it might have or it can have the effects on the endocrine disruption potential. It may either act as an anti-androgen or estrogenic activity. So that we proved in the in vivo approach also, where we noticed the expression profile of AR genes, ESR gene, and the vitellogenin genes were significantly increased. Particularly if you see the AR gene and the ESR gene, the expression is not uniform, dose dependent. Usually you may ask in pharmacology and toxicology, the dose response should be, as the concentration increases, obviously the response should also increase. But in this case, it is not happening. Why? This particular response is called as a non-monotonic dose response, NM non-monotonic dose, NMDR. This NMDR is a characteristic feature of endocrine disrupting chemicals. Endocrine disrupting chemicals will not give you a clear-cut dose response. It has its efficacy. It has this at a higher concentration, it may reduce the response, where at low middle concentration, it may enhance the response like that. So this is clearly we have shown in our Western blot analysis also. This Western blot analysis clearly, if you see, instead of 550, 500, 5 is increased, 50 is again increased, but 500 it is decreased. So this shows that this 50 ppb is the optimum concentration of this particular chemical contaminant inducing the endocrine activity, disruption activity in the zebrafish. So this is clearly seen from our Western blot results also, where that 50 you can see the band is very intensity. The intensity of the band is very high compared to the 5 and 500. So that the expression is statistically significant with respect to the control. So this clearly shows that yes, this particular compound is having the endocrine disruption activity, which is proven from both the transcriptional and the translational targets. So now the adult zebrafish, which we raised up to 6 months, we have taken out and we studied the survival. How many were survived? So there is a significant survival decreased in the highest concentrations. But however, you can see number-wise it is decreased, but statistically it is not decreased. And another question, another important thing is in control we have only 30%. You may ask why only 30% only in the controls in a 6 months? Because see, this is not unlike like higher animals like rat or mice, because this maintenance is very easy only. But still, these are all water-based organisms. The metamorphosis, that is developing stage from one to the other, the mortality is unavoidable. So this 30% is something allowable in particularly in the zebrafish. Comparing to that 30%, the treated ones have a significant decline in their survival. But however, we didn't find any differences in the number of eggs laid by the male and the female groups. The clutch size is not different. Though they are arbitrarily, you can see the less numbers, but they are not statistically significant. But however, we now did the fertilization rate. So we performed both interbreeding and the intrabreeding. That is, we taken the male fishes from the control group and female fishes from the toxicant exposed group, and female fishes from the control and male fishes from the toxicant. So this is called as the interbreeding. So when we do the interbreeding, we didn't find any significant effects on the fertilization rate. They are absolutely fine. But however, among the group, that is the intrabreeding, where male and female from the treated ones, we have seen the fertility, the eggs are not fertilized. We have got only lesser than 60% of the fertilization rate, which is significant. So it shows that this compound has impact on the fertilization rate when the animals grow into the adult stage. The effects, we have exposed it only in the 4 days only. But we have the effect is still persisting on the 180 days only. It should looks like the events are etched in the organism in the early childhood itself. That is the early stage itself. So then what we did again, we made the exposed animals to get the F1 progenies. Whether F1 progenies is also having any developmental defects or not as such. Yes, we have seen the same axial curvature is induced in the F1 progeny also. And next we see the survival of these F1 progenies that is also significantly decreased when compared to the control ones. So at the end, we can say the effects on the ESR, VTG, and the AR gene and the proteins expression indicated the endocrine disruption of this particular 4-methylbenzylidene camphor. This eventually long-term maintenance affects the survivors at higher concentrations with no significant changes in the clutch size. However, there is changes in the reproductive gene and protein expression indicating that 4-MB is reprotoxic. So besides, it induced developmental defects and reduced survival of the F1 progenies also. So the summary is the zebrafish embryos and larvae, we have studied development, nervous system, and the reproduction with the different changes in their various endpoints, inducing developmental toxicity, neurobehavioral toxicity, and reproductive toxicity. So a tiny fish with definitely a big splash in different domains of the toxicology using this particular model compound. But still, now you may ask, you may have one question in your mind. This fellow is telling keep on what is the advantage and everything. Don't it have any limitations of this model? Because each and every model have its own advantages and disadvantages also. Likewise, this particular model system has its own limitations. For example, these are all short-lived animals. The maximum lifespan of this one single zebrafish is around 4.5 to 5.5 years only. So you cannot directly compare with the human. The direct comparison of age-related phenotypes are limited with this particular model system. Then second one, they have simpler organs when compared to other mammals which have complex. So you cannot able to directly correlate the physiological functional perspective because some organs are not conserved. For example, zebrafish lacks mammary glands, lungs, and prostate glands. So you cannot, if a researcher wants to study the effects on these organs related to any of the functional perspective, this model is not a suitable model. And another thing is the genome size. Its genome size is 1/2 of than the human genome. So the making the direct comparison is difficult. So then what else it will suffice? Yeah, this can be used as a screening tool before you are taking into the higher animal model. And to certain extent, you can extrapolate it, but you cannot have a direct extrapolation to the human health. But still, this has a translational value. What is the translational value? One is you can assess the toxicity of chemicals and drugs across the lifespan because of a rapid development. And second thing, you can elucidate the chemical mode of action as I presented earlier. The next, you can also develop or test some of the interventions based on your behavioral endpoints. Because like Professor Red said, ethanol, when you expose the ethanol, zebrafish also behave like a human. You can see how it is, the behavior, their motion, their orientation, everything will be, you can easily study it in the using the ethanol after ethanol exposure. And similarly, you can predict the chemical toxicity into the other species. So these are some of the translational value of this model in the area of toxicology. So with this, I will end my talk. And here I can say what the zebrafish is telling to us. Any guess? I am the gatekeeper for you. It is a gatekeeper for us. So it is a screening. We can use it as a screening model before whatever the n number of molecules will come to the market so that we can use it on the other animal models, higher animal models before it reaches the humans. So thank you so much for your patient listening. And thank you for this opportunity given by the organizers. And really, I'm thankful to Professor Kamar Rahman for this giving me this opportunity. And she is the living scientific legend, which for this adage sky has no limit. So we should give a big applause for her. Because I never worked with ma'am, but I heard about ma'am. I have seen a lot of her outputs that she carried out when she was associated with IITR, but still she's our family member only. I have seen her in a lot of our functions. but still, yeah, she is a scientific living legend for this particular thing. Sky has no limit, where we don't have a depth of knowledge of learning. So she's one of the best example for it. And really, I'm thankful, madam, for giving me this opportunity to deliver my piece of research work in front of you all. Thank you so much.
Thank you, sir, for this elaborate session. I am quite sure that this particular session will be of definitely helpful to many of our PhD scholars and researchers working in this particular field. And we have got so many questions online, so it will take more time for you to addressing each of them. Hope will take almost every one of them. But I could find some couple of questions from one of our attendees named Shivani Pandey. The first question is that she's asking, as we know there are so many type of database models, so many identical patterns. If after testing we achieve a good LD50 range, but that particular variety uses its own identical body pattern, only not their same or any other body damage. How will we conclude our results, whether we are succeeded in that testing or not?
Can I?
Yes.
See the contour again? Which one?
So this.
This one.
Yeah.
So one thing is, it is again depend on the which type of variety you are using. For example, there are so many varieties, but the most widely used strain in the zebrafish is the ASWT, that is the adult Assam wild type strain. It is one of the most widely used because there are so many nowadays. Fluorescent strains are also available, like orange color is there, green color zebrafish. These are all also there. But for the research, the most widely used till date using is the gray silver line stripe based zebrafish, which is called as the adult wild type zebrafish screening. So if you derive that results from that particular thing, I think that will be of more what you can say, that will give you a more confident thing. And another thing you mentioned, the LD15, you have to correct it as a LC50. It is not in case of zebrafish. If you are deriving the results, don't mention it as LD50. You have to mention this as a LC50. So, but however, since you use the other thing, maybe you can do one thing, you can just have a cross verification with this particular model and you can see the comparable results if you get a unique result between these two models, and you can go ahead with that. Because always there expects a biological variation will be there when you are conducting among the variety also.
So I do hope that the answer is reached to her. The next question is also from Shivani Pandey, sir. She's asking that I need to ask if all parameters comes under the acceptance range, but that variety uses its own pattern. So will we consider that the particular drug molecule is good enough?
So what kind of pattern it is using, that I'm not getting it, and what kind of parameters you have studied, that also a bit unclear here.
So I do suggest that Shivani will forward your queries to sir in his mail ID, and sir will get back to you in detail. The next question from Kaushik is saying, she's asking the physical working on script is more appropriate than software working, or which can give the best result while comparing?
See, there is nothing like this can be replaced 100% by that thing. It is like a balance. You have to be in a balance which it will complement each other. For example, all fine day you cannot rely only on the docking or the computational or the AA and the ML based approaches. The AL and ML based approaches, everything will be give you some sort of a filtering out so that you can able to screen out. I mean, you can able to remove some of the unwanted things based on these particular software. So you can finally end up from 1000 to 100, 100 to 10. So those 10, again, you can go into your molecular docking. You can find out which is your target. For example, if you are targeting a particular ADME pattern, or if you're targeting endocrine disruption, if you're targeting cancer, then that particular target, if you want to do a study based on your computational tools, those findings you can cross check with your in vivo, and so that it can be both the results will complement to each other. So in that case, both have its own weightage. It's nothing like one is lower grade and other one is a higher grade. Both are having equal weightage and both needs to go parallelly. It has to go. That is how even the regulators and also the other agencies used to recommend it. Apart from that, there is now one more thing is coming to the regulatory arena like this adverse outcome pathway, AOP network, which is based on your these findings only that AOP network will come into the picture. So it looks like both are having equal weightage and both will be essential in terms of the risk assessment.
Thank you, sir. We have one query from our own student from County here, 15th annual year, and she's asking that what is the age limit of children? I mean the lifespan.
Yeah, lifespan, as I mentioned, adult lifespan in the can live up to 4.5 to 5.5 years. But the ideal, for example, if you want to study the reproductive effects and all, after 1.5 hour, after 1.5 year, you can use the maximum 2 years, you can able to use the breeding pairs for getting the embryos, because afterwards the quality of the eggs will be getting declined as the age progresses. So that is a well-known thing. It is not only related for the higher animals, even if it for the lower animals also. So though the lifespan is 5.5 years, the ideal time which you can able to work on the embryos, you want to create a transgenic lines, or if you want to work on your development on the reproduction is between 1.5 to 2 years. That will be the ideal, because they reach the sexual maturity in 3 months. 3 months may they reach the sexual maturity. Some 4th month onwards, they are ready to get the eggs. So that time onwards, you can start your experiments and you can get the embryos for your research work.
A query from one of our attendees, his name is not clear over here. He's asking that which are the software that are used for recording and analyzing activity of embryos.
This is not like a specific software. It's again depending on the user, because this is always the softwares will be associated with the microscope you are using it. For example, there is one particular thing, Noldus Technologies in Netherlands, where they have a separate software exclusively for the zebrafish associated. It is compatible with their microscopes. So like that. So they, apart from that, there are other individual equipments are available to study the behavioral assays. Small animal behavioring equipments are available, which you can able to study the n number of behavioral patterns for both your toxicological aspects also and for your pharmacological aspects also, that you can have it.
Okay, we are still having several queries floating on online platform. So I do promise that the mail IDs of both our speakers will be there at the end of the ceremony, and you can put forward your queries on the mail. Sir, I have one query, no, may not be a query, but a suggestion that for establishing a new geographic lab setup, what all requirements are required, and can you just highlight on the cost parameters?
Yeah. The first and the foremost requirement is the funding. Okay. So it's up to the fund this thing. So as I mentioned, you can start from the glass aquarium onwards. You can establish this, because the glass aquarium disadvantage is because I started it from the glass aquarium. The only thing is handling is an important thing, because we should not induce stress to the animal, which will eventually affect our results on data results. And daily you have to clean it, and daily you have to change the water. So you need a dedicated lab assistant for it. So if you have good amount of funding, you can go through a zebrafish standalone system is currently available. So it is available in both in India is also available, and it is also importers are also there. Currently, we are using in our lab is the Zeptec standalone system by Techniplast Italy. So we got it in 2017. That time it was around 17 lakhs. Now it is coming around 30 lakhs. So it is a rack-based thing where the water will continuously flow into this, so that you will no need to change it, you no need to disturb it. Only thing you have to feed it. That system will automatically adjust the parameters such as water temperature, pH, conductivity, everything. And it also having a automatic dosing unit also. Whenever there is a change in the pH or change in the conductivity, it will automatically suck the salts and it will release into the water, so that it will maintain it. And it is having the alarm also, which you can connect to your mobile. It will tell at this particular time the power is off, the instrument, this parameter is missing out. So you can have the data logger facility also. So that way you can able to minimize the entry of this maintenance stress you can minimize the stress by maintaining this particular unit. So this one unit we are having, which we are doing both breeding also, we are maintaining our own stock also, so that anytime the students can, because the problem here is these are all water-based animals. We cannot get embryos from all the pairs. So if we keep 10 pairs of zebrafish for breeding, we will get only 4 or 5. That itself is a very successful rate. So 50% of the breeding pairs will give you embryos. That itself is good enough, because if from a single breeding pair you will get 400 embryos. So obviously you may need only 150 to 200 embryos for your experiments, including your triplicates. But still you can conduct this same, repeat the experiments in 3 times, because nowadays repeatability is more important to prove our results. That is more important. So in that case, this is very important thing. This model is highly useful.
Thank you, sir, for this informative session. And last year we have our M-Pharmacy students, those who have visited your labs and continue with their training programs. And I would request you to kindly inform us, keep on informing whenever you are planning for such programs. This time also we will inform our students. We have a good lot of students with that, so we'll definitely plan for
Sure,
the
sure,
meeting.
sure, sure.
Okay, so thank you, thank
So,
you.
Thanks,
Thank
thank
you
you.
for joining us.
Thank you.
Now I request Sriviki to present over the meeting. Thank you, ma'am. As we have reached the end of inaugural ceremony of International Conference on Workshop AAE 2023, I would request Dr. Lucy Mohapatra, ma'am, the Organizing Secretary of AAE 2023, to offer the vote of thanks. May I request my organizing co-mates, Dr. Yasin sir, Dr. Shraddha ma'am, and Dr. Rahul sir, to please be on stage with me while I am offering this vote of thanks. So, thank you is such a prayer that cannot be seen or touched. It must be felt by heart. I stand here today as one of the proud and grateful organizing secretaries, along with my fellow mates, Dr. Shraddha Bist, Dr. Rahul Kumar Warrior, and Dr. Mohammad Yasir, to express my heartfelt thanks to all those who have made this event possible. I would like to start by thanking our own beloved Director AIPAU Lucknow, Professor Dr. Anuradha Mishra ma'am, who have always been supportive of our initiatives and encouraged us to strive for excellence ever since he conceptualized this grand 3-day long event on hybrid mode. Words are not enough to thank you, ma'am, for your constant guidance and support to take this international conference works on this great standard. Thank you, ma'am. I thank all the honorable dignitaries, including esteemed Pro Vice Chancellor, Sir, Dean Academic Sir, Kamal ma'am, and honorable speakers, Dr. S.K. Rath, Dr. S. Anjumani Sir, and the other HOIs for taking out time of their extremely stringent schedule and grace this inaugural ceremony with their presence. So thank you all. An event of this dimension cannot happen overnight. The wheels start rolling months in advance. We have been fortunate enough to be backed by our organizing team of very motivated and dedicated faculty, students, volunteers. I thank everyone enough for the involvement they have shown and the willingness they have expressed to take on the completion of the task beyond their comfort zones, even during the weekends. The ID team deserves a special thanks for looking into the proper conveyance of this program online. So thank you, Gaurav Mathur sir, Pragyan sir, Pragya ma'am for the same. Special thanks to S.K. Sahuji for capturing these wonderful moments and making it a memory. And I thank all the attendees attending this event online and all our students of pharmacy and pharmacy attending this offline. We have also couple of registration, on spot registrations today, and the students are also joining us. So I thank you them also for joining this event, and I thank you all of you for being the part of this particular event. Thank you. I would request now everyone to stand up for the national anthem. Biswajit. So with this, we come to the end of the inaugural ceremony of day one. I request the honorable guests and speakers to join us for the lunch at the VIP norm, and I request Shraddha ma'am to kindly escort our esteemed dignitaries for lunch at the VIP norm. I request all our attendees online and offline to join the technical session 3 at 2:30 p.m. by Dr. Tavinda Hassan, Associate Professor of Human Anatomy, Histology and Embryology, College of Medicine, Prince Noor bin Abdul Rahman University, the Kingdom of Saudi Arabia, on neuronal stem cells, a contemporary world for disease and modeling, and therapeutics in regenerative neuroscience, and an emerging alternative to animal models. So the link has been already shared with you all previously. I request you all to join us sharp by 2:15 p.m. so that we shall be able to start the session on time. I request Dr. Pragya ma'am to kindly re-share the link in the WhatsApp group. Thank you.